So I will try to be reasonably concise. Energy-bearing nutrients: proteins, carbohydrates, lipids. Vitamins and micro elements are essential, but have no energy value. Now, carbohydrate digestion. You have to remember that carbohydrates digest to monosaccharides. This three: glucose, galactose, fructose. Glucose can be derived from virtually any sugar-containing food. Galactose, milk. Fructose is derived from specifically sugar. Okay, does that make sense? Processed food contributes so much to obesity epidemics in the US. It contains a lot of simple carbohydrates. End products of the fat digestion: fatty acids and glycerol. Saturated versus non-saturated fats. What you need to know: saturated fats are solid, unsaturated are liquid. Saturated are from the animal source, unsaturated from the plant source. Main exogenous sources of cholesterol. I'm not going to ask you to list me the foods that rich in cholesterol. What I want you to understand very clearly is that the majority of cholesterol is produced endogenously, meaning produced in the body. We do not receive a majority of cholesterol with the food, only about 15%. Does that make sense? Saturated fats, unsaturated fats, and cardiovascular disease. So, dietary saturated fats do not increase the risk of cardiovascular disease. Unsaturated fats seem to decrease the risk of cardiovascular disease. Does that make sense? This is what I want you to understand, that dietary saturated fats in general population are not associated with elevated risk of cardiovascular disease. Trans fats, chemically modified unsaturated fats to turn oils into the solid. Relationship: trans fat consumption increases the risk of cardiovascular disease. We're good so far. Protein digestion to amino acids. Essential amino acids must be provided with the food. Non-essential amino acids can be synthesized. Essential amino acids can be found in any animal protein. Vegetarians, as we just discussed, have to, if they're strict vegetarians, legumes plus vegetables or greens. Metabolic consequences of long-term high-protein diet. Acidification of the blood. Okay. If you strictly eating, just meat, then it can be not beneficial to the bones. But generally, we receive enough calcium with the diet. All non-rule in relation to proteins in the diet, meaning that in order to synthesize proteins, all 21 amino acids must be present. Are we clear? Positive nitrogen balance is when protein is deposited. Positive nitrogen balance is characteristic for kids that are growing, okay, or athletes that build muscle mass. Negative nitrogen balance is when protein, muscle protein, is catabolized, broken down for energy. This is characteristic for starvation. You need to use carbohydrates for the proper utilization of the protein for muscle growth. If carbohydrates are absent, proteins will be used as a source of energy, and it will slow down or completely stop muscle growth. Vitamins are organic compounds that often serve as the cofactors of different enzymes. Two major groups are lipid and water-soluble vitamins. Lipid-soluble vitamins, in terms of intestinal absorption, they require fat consumption. They can be absorbed only with fats. And interesting feature of fat-soluble vitamins, they can accumulate in the body. Water-soluble, excess of water-soluble vitamins will be excreted with the urine. This for fat-soluble vitamins, you have to know. Now, Marina asks today about function. What do you need to know about the function? Please do not memorize which enzyme they are cofactor for. You need to know the dis function. So I said for A, night blindness. For D, bone problems like rickets in children or osteomalacia and osteoporosis in adults. No question about vitamin E. Vitamin K, clotting problems. B1, beriberi, the neurological disease associated with the deficit of this vitamin. Deficit of vitamin B12 leads to pernicious anemia. Okay? Deficiency of vitamin C leads to scurvy. Okay? Pathologies of the skin, hair, nails, so on. So far, am I clear? This is now, as I mentioned before, I cannot ask you what are the problems associated with the deficit of vitamin B6. I can't. But I can ask you which of the following vitamins are lipid soluble or water soluble. And I can include any vitamin in the list. So you have to know which ones are water soluble, which ones are lipid soluble, which is super simple. As long as you know 4 lipid soluble, A, D, E, and K, everything else is water soluble. Am I clear? Macroelements. Focus on sodium, potassium chloride, calcium, and phosphate. That's it. Deficit of calcium and phosphorus lead to the bone problems. Sodium, potassium, and chloride lead to the problems with the neurological problems and cardiovascular problems, inability of a heart to contract, or poor transmission of the nerve signal. Electrolyte imbalance. Iron deficiency. Now, among microelements, iron, fluorine, iodine, and zinc. Iron deficit, anemia, fluorine deficit, caries, iodine deficit, hypothyroidism. We're clear. Metabolism, anabolism. Metabolism breakdown, anabolism synthesis. Exergonic is energy releasing reaction, endergonic, energy requiring. Well, terrible question. Why is the efficiency of energy transformation is less than 100%? Well, because that's how chemistry works. The difference, so the efficiency of every process is less than 100%. The difference between the energy produced in one reaction and energy consumed in another goes towards heating the body. So glucose in the process of in the process of its catabolism, glucose is oxidized to carbon dioxide and water. Oxidation of glucose in the presence of oxygen, we call it aerobic respiration. Without oxygen, it's fermentation. Final electron acceptors. In aerobic respiration, it's oxygen. In fermentation, it's pyruvate. Are we clear so far? The electrons in the cell are carried by molecules, 2 molecules that we discussed, NADH. Well, okay, pair. NAD plus, NADH, and FAD, FADH2. Regarding these pairs, you have to know which form is reduced, which form is oxidized. Am I clear? Any questions on this? Remember, more hydrogens, it's a reduced form. So NADH, FADH2 are reduced form. 3 major steps in the glucose metabolism, the ones that we just saw in the quiz: glycolysis, Krebs cycle, and oxidative phosphorylation. In glycolysis, glucose is completely oxidized. Not completely, sorry. In glycolysis, glucose is converted to pyruvates, and it happens in the cytoplasm. Okay? Key phosphorylate 3-carbon intermediate is G3P, or glyceraldehyde 3-phosphate. And the final products of glycolysis are pyruvate, 2 molecules of ATP, and 2 molecules of NADH, carrying electrons. Sorry, disregard that second one. It asks about final products of glycolysis twice. So in the presence of oxygen, pyruvate enters the transition step. In the absence of oxygen, pyruvate is reduced to lactate, or lactic acid. And this is the fermentation. We clear?
What did you say for what happens to pyruvate in the
Presence of oxygen, it enters the transition step. So during the transition step, pyruvate is oxidized to acetyl-CoA, and a molecule of CO2 is leaving the party. 1 molecule of NADH is produced. Okay. Transition step and Krebs cycle happen in the mitochondria. Acetyl-CoA reacts with the molecule of citric acid, so 2 carbons of acetyl-CoA react with 4 carbons of oxaloacetate, forming citric acid. So citric acid is formed, 6 carbons are in this molecule. Citric acid is converted to alpha-ketoglutaric acid. Since alpha-ketoglutaric acid has only 5 carbons, and we go from C6 to C5, that carbon that is lost, sort of, leaves the reaction in the form of CO2, that's the second CO2 molecule, and NADH is produced. Alpha-ketoglutaric acid is oxidized to succinyl-CoA, which is 4 carbons, another CO2 is produced, okay? Does that make sense? And another NADH. By the time we reach succinyl-CoA, we completely oxidized all carbons in the molecule of glucose. Are we clear? Now, this question, succinyl-CoA eventually is converted to oxaloacetate, or oxaloacetic acid. And during these reactions, in a Krebs cycle, 1 molecule of ATP is produced, 1 molecule of FADH2 is produced, and 1 molecule of NADH. We're clear? Okay. So during Krebs cycle, all carbons of glucose molecule are oxidized, completely oxidized, to carbon dioxide. Does that make sense? Okay. So far, before we move on, any questions? So summary of products for 1 Krebs cycle. For 1 Krebs cycle. Now that can come a little confusing. So let me ask you this: do you want the summary for 1 Krebs or for 1 molecule of glucose? It doesn't really matter for me. Let's do both, okay? So for 1 Krebs, 4 NADH, Krebs plus transition step. 4 NADH, FADH2, ATP, CO2, 3 CO2s. Now, if we want to say what is the outcome of transitional step in Krebs cycle for the entire molecule of glucose, what should we do with these numbers? Double it. Eight NADH, 2 FADH2. Well, you can continue. So how many Krebs cycles are necessary to oxidize one molecule of glucose? 2. Good. Now, oxidation and reduction. Oxidation and reduction. Oxidant is the one that accepts electrons. It takes electrons from another molecule. Reductant is the one that gives electrons. So oxidant is reduced, reductant is oxidized. When molecule is reduced, it gains electrons. Are we clear? When molecule is reduced, it gains electrons. When molecule is oxidized, it loses electrons. Okay? When molecule is reduced, it gains electrons. When molecule is oxidized, it loses electrons. NADH and FADH2 are electron carriers. No, they're reduced in oxidized forms. I hope you do. Final destination for NADH and FADH2 is electron transport chain, and they deliver electrons there. Proteins of electron transport chain use the energy of electrons delivered by NADH and FADH2 and pump hydrogen ions across the mitochondrial membrane, creating electrochemical gradient. Since hydrogen ions are charged and hydrophilic, they cannot directly diffuse across the membrane in mitochondria, but are allowed to cross the membrane via ATP synthase. As they diffuse, or as we call this process, chemiosmosis, they perform chemiosmosis. As they diffuse through ATP synthase, the kinetic energy of the hydrogen ions movement provides the energy necessary for ATP synthesis. So ATP synthase essentially converts kinetic energy of moving hydrogen ions into the chemical energy of the bonds in the molecule of ATP. We clear? In the meantime, electrons are accepted by oxygen, which is the final electron acceptor in the anaerobic respiration. Hydrogens join oxygen as well, producing water. So chemical summary of glucose oxidation will be molecule of glucose interacts with 6 molecules of oxygen, producing 6 molecules of CO2 and 6 molecules of water. I want to be very clear that you will not have to write down any of the reactions. What you need to understand, or you need to be able to say, that final products of glucose oxidation are carbon dioxide and water. Am I clear? Now in fermentation, the final electron acceptor is pyruvate. It happens in the cytoplasm. And the final ATP numbers for oxidative respiration. Again, if you don't remember whether it's 34 or 38, it is totally fine. I'm not going to ask you this closed numbers. Does that make sense? You have to understand that it's in a ballpark of 30 plus ATP. It's not 40 something. It's not 20 something. Am I clear? Okay. Now, for fermentation, 1 molecule of glucose will give you just 2 molecules of ATP. Cells cannot store ATP for a long time. So when ATP levels in the cell increase, cellular respiration slows down, and all excessive glucose will be stored as a glycogen in the process of glycogenesis. Okay? So when we start fasting, glycogen goes first in the process that is opposite to glycogenesis, called glycogenolysis, post-absorptive state. So when glycogen stores are exhausted but glucose is still necessary to fuel up the brain, okay, brain is metabolically relying on glucose, glucose will be generated in the process called gluconeogenesis. Now what about lipids? Turns out lipids store more energy per molecule than carbohydrates. And when glycogen storage is exhausted, the source of energy, the primary source of energy, will be triglycerides. They will be broken down in the process of lipolysis, and the first step will be to break down triglycerides to glycerol and fatty acids. Glycerol will be converted into glyceraldehyde 3-phosphate, essentially entering glycolysis. Free fatty acids will be catabolized in the process of beta oxidation, which occurs in mitochondria. Product of beta oxidation is acetyl-CoA. Acetyl-CoA enters Krebs cycle, and then, all other steps are all the same. Now, what if you have a situation opposite to starvation, and you have a lot of excessive glucose, and all glycogen storage capacity is filled? In this case, excessive glucose, products of glycolysis, will be used for synthesis of glycerol and fatty acids. Glyceraldehyde 3-phosphate will be converted to glycerol. Glyceraldehyde 3-phosphate will be converted to glycerol, and acetyl-CoA will be converted to fatty acids. Glycerol and fatty acids will combine in the process called lipogenesis, producing triglycerides. And triglycerides are stored in adipose tissue. So we just explained glycerol comes from glyceraldehyde 3-phosphate, and fatty acids are synthesized from acetyl-CoA. Now in ketogenic diet, where glucose is low, the process of gluconeogenesis uses oxaloacetic acid for the synthesis of glucose. When oxaloacetic acid is used for glucose synthesis, the Krebs cycle is essentially shut down. In this case, acetyl-CoA has nowhere to enter, and it is converted into it's converted into ketone bodies in the liver. Ketone bodies are used as the main source of energy by virtually all cells of the body, sparing glucose for the brain. So, obviously, in case of low carbohydrate, high fat diet, this type of diet will provide better outcomes in terms of the weight loss compared to high carb, low fat diet. Because when you eat fat, you burn fat. Does that make sense? Now, high fat, low carbohydrate diet will also reduce insulin levels, increasing insulin sensitivity, and it will help to increase the percentage of lean body mass. Now, cholesterol in the body is mostly used for the synthesis of the steroid hormones. And different chemical mediators. It's mainly coming from the saturated fats. Saturated fats endogenously are converted into cholesterol. So the majority, 85% of cholesterol that is used in the human body is produced inside of the human body. It does not come with the diet. It's transported in the blood in the form of lipoproteins. Low and very low density lipoproteins carry cholesterol from liver to tissues. Not only cholesterol, but also all other fats. Does that make sense? From tissues to the liver, cholesterol and fats are carried by high-density lipoproteins. So, the total levels of cholesterol do not accurately predict the risk of cardiovascular disease. What actually predicts the risk of cardiovascular disease better is the ratio between LDL and HDL. So the ratio between LDL and HDL should be as low as possible, meaning that LDL should stay low and HDL should stay high. The higher levels of high-density lipoprotein and lower levels of low-density lipoprotein are associated with a decreased risk of cardiovascular disease, and vice versa. You can remember it this way. Low-density lipoproteins are really low, they're really bad, and they increase the risk of cardiovascular disease. High-density lipoprotein are high quality, and they decrease the risk of cardiovascular disease. We clear? Now, effects of saturated fats on cholesterol synthesis. Generally speaking, elevated levels of saturated fats increase cholesterol synthesis, but in the general population, consumption of saturated fats do not alter blood cholesterol. Again, if you decide to take up the butter diet and eat butter, only butter and nothing but butter, then yes, your cholesterol levels eventually will skyrocket. Unsaturated fats generally decrease the cholesterol levels in the blood, while trans fats undoubtedly increase. Dietary cholesterol and plasma HDL-LDL ratio have very little connection. Dietary cholesterol, you understand what dietary cholesterol means? Cholesterol that you get from the diet. So, the amount of cholesterol that you consume with food isn't really influencing the HDL-LDL ratio. Now, two main catabolic reactions in which proteins can be used as the energy source are transamination and deamination. In transamination, keto acids are synthesized. In deamination, amino group of amino acids is removed. Eventually, it is converted into ammonia. Ammonia is excreted in the form of urea, the urine. Products of deamination and transamination. I should have wrote two, deamination and transamination. Products of protein catabolism can enter glycolysis and Krebs cycle. Does that make sense? Do you understand what I'm saying? Now, you see this one. So we have this scheme behind my back. That's one. So that's the first sentence. First sentence is drawn on the whiteboard behind my back. Are we clear? Now, what about second sentence? Metabolic changes associated with the dietary changes. Don't freak out, please. What you need to know is, I tell you, for instance, hypothetically, you have a diet that is extremely rich in carbohydrates. What's going to happen? You can tell me. Glycogen is going to accumulate. Can you see that behind my back? Glycogen is going to accumulate if there's a lot of glucose, and fat is going to be produced. Does that make sense? Tell you, okay, now we have a diet that is really high in lipids. Triglycerides will be catabolized to fatty acids, and glycerol will enter glycolysis, fatty acids will be oxidized to acetyl-CoA. Tell you, okay, next, you're going to have a diet that consists like 90% of your diet is protein. So what's going to happen? Protein will be used as the main source of energy. It will be deaminated, transaminated. The products will enter the glycolysis and the Krebs cycle. Are we clear? To that extent. I'm not going to ask you a complicated question, something like you have a diet that, I don't know, 40% protein, 30% lipid, 30% carbohydrates. What's going to happen? Don't worry about it. Am I clear? I have no idea what's going to happen. Okay. Now, a few last things. Absorptive state. After the meal, anabolism dominates. Monosaccharides in the liver are all converted to glucose if they aren't. And glucose will be used as the primary energy source, okay, in glycolysis. Excess glucose will be converted into fat, and dietary lipids will be stored in the adipose tissue. Amino acids are taken into the cells, and, well, are they used as the energy source? Well, some of them, yes, but mostly amino acids will be used for protein synthesis. Does that make sense? In absorptive state, protein synthesis will be dominant. Insulin regulates absorptive state the most. What triggers insulin production? Let's recall, What? Well, glucose and amino acids, right? Elevated blood glucose, elevated blood amino acids. We're clear. So, you see the cellular metabolic processes are upregulated. The table in the lab, work with it. Be able to answer it without looking. It will really help. And don't just memorize. Understand why it happens. Are we clear? Post-absorptive state. After the meal, metabolic purpose of post-absorptive state is to maintain stable levels of glucose in the blood so brain can function. In liver and skeletal muscles, glycogen is broken down into glucose. In adipose tissue, triglycerides broken into fatty acids and glycerol with subsequent effects. Proteins are catabolized to amino acids, and amino acids are used as the energy source. First, muscle proteins let go. Does that make sense? Your muscles will be used first as the source of energy. Kidneys participate in gluconeogenesis, while the majority of the body tissues switch to ketone bodies as the primary source of energy. So glucose can be spared for the brain. Brain can use ketone bodies as the source of energy, but it takes a lot of time. Glucagon regulates post-absorptive state. Again, this story, you see this last one? Which cellular metabolic processes? Table in the quiz. Well, inhibited. So, hypothalamus controls the food intake, right? Ghrelin, glucagon, and norepinephrine increase the hunger feeling. Leptin and insulin inhibit appetite. Elevated levels of amino acids and glucose, they inhibit appetite. BMR is the amount of energy that you spend by not doing anything. The basal level. Okay, body surface. Higher body surface, higher BMR. Older age, lower BMR. Female gender, lower BMR. Higher body temperature, higher BMR. Stress, elevated BMR. Thyroid hormone, elevated BMR. Elevated muscle mass, exercise, alcohol, protein-rich food, all increase BMR. Body temperature is controlled by hypothalamus. Core temperature inside, shell temperature outside. Okay, cooling the body. You know what cools the body, what increases body temperature, okay? We're good? Sweating versus shivering. Heat stroke. Overheating turns off the protective mechanisms by hypothalamus. You heat up, your BMR increases, that automatically increases your body temperature, that increases BMR, positive feedback. Hypothermia, the opposite. You cool down, hypothalamus turns off, your BMR decreases, you cools down even further, and that goes and goes and goes. Fever, hypothalamic setting of the normal temperature is reset at the higher number. So instead of 37 degrees Celsius, your normal now is 38 or 39. You start shivering and become pale to increase your body temperature. That make sense? Fever restricts the replication of the pathogens and helps to sequester iron to reduce the pathogen replication.